Today, we're going to talk about something that is totally changing our understanding of human biology. It's called multi-omics, and it's all about moving from looking at just one tiny piece of our biological puzzle to finally seeing the whole incredible picture.
I mean, just think about that question for a second. It sounds like something straight out of a sci-fi movie, right? But this is the real-world promise of precision medicine, the idea of creating treatments that are perfectly tailored to you. And making that happen all comes down to integrating just massive amounts of data.
So, what is this multi-omics thing anyway? Well, think of your body as this incredibly complex system with a bunch of different layers. You've got your genome, which is kind of like your body's DNA blueprint. Then there's the transcriptome. That's the messages being sent out from that blueprint. And then you have the proteome, which are the proteins that actually go out and do all the work. Multi-omics is all about studying these layers, all these omes, together, at the same time to get that big, holistic picture.
Now, to get that big picture view, you first need the data. And in the last decade or so, we have gone from a tiny trickle of biological information to an absolute fire hose.
This quote here really gets to the heart of it. Brand new technologies now let us measure pretty much everything, from our genes to our proteins, on a massive scale. To put it in perspective, we can now get more data from one single biological sample than we could have gotten from thousands of them just a few years ago. And that creates this incredible opportunity, but also a massive, massive challenge. And that challenge is really what we're going to unpack here. You see, having all the pieces of a puzzle is one thing, but actually putting them together so they make sense, that's a whole different ballgame.
This visual right here just lays out the problem perfectly. Imagine you've got separate puzzle boxes for each data type, one for genes, one for proteins, and so on. As you can see on the left, you're trying to build a picture of different patient groups. But the data from the different boxes, it doesn't always line up. And even worse, there are often these huge gaps where entire sections of the puzzle are just gone. So the goal, the really crucial step here, is to use computational tools to solve this mess before we can even think about finding biological clues. And as you can see on the right, that means a few key things. We have to reduce the mind-boggling complexity, correct for any errors, and, this is the big one, we have to do something called imputation, which is really just a fancy way of saying we need to intelligently fill in those missing pieces. So let's just quickly break down the three biggest technical headaches. First, you've got high dimensionality. That just means we have thousands and thousands of data points for every single person. Second, heterogeneity. This means the data for genes and proteins just look different. They play by different rules. And third, sparsity. Missing data isn't just a possibility, it's everywhere. It's the rule, not the exception.
Okay, so faced with this ridiculously complex puzzle, where did scientists start? Well, they turned to the statistical tools they already had. We can think of these as the classical or old-school methods.
The most straightforward way to start is just to look for simple connections. Methods like Canonical Correlation Analysis, or CCA, which you see in this first diagram. They're essentially trying to draw straight lines between data points in one set and data points in another, just to find the strongest, most direct links.
Another classic approach, which you can see in the second diagram, is something called Matrix Factorization. This is a really powerful way to cut through the noise. It works by finding a much smaller number of shared underlying patterns or factors that can explain most of what's going on in the data.
And a third strategy, shown over in diagram E, is super clever because it focuses on the patients themselves. These network-based methods build a who's similar to whom map for each type of data. Then they fuse all those maps together, making the connections that show up over and over again stronger. It's a great way to find really solid groups of similar patients.
But here's the catch, and it's a big one. Biology is almost never simple or linear. It's messy. These classical tools are good, but they're built to find straight-line relationships. They really struggle to pick up on the incredibly complex, subtle, and non-linear patterns that are actually running the show in our bodies. It was time for a new approach.
And that brings us to the deep learning revolution. All of a sudden, scientists realized they could apply the same kind of advanced AI that powers things like image recognition and language translation to this huge multi-omics puzzle.
This table really makes the difference pop. Where the classical methods were mostly stuck looking for simple linear patterns, deep learning models are amazing at learning really complex, non-linear relationships. But the truly game-changing part is that second column, generative power. This is the ability not just to analyze data, but to create new data.
Let's see how that works.
Okay, so let's look at one of the fundamental tools in this new AI toolkit, the autoencoder. Here's the idea. An encoder network takes in all that messy, high-dimensional data and squeezes it down into its most essential information. That super compressed summary is called the latent space. Then, a decoder network takes that compressed code and tries to rebuild the original data perfectly. That latent space is the magic. It's this pure, distilled summary of the most important patterns.
Now, this leads us to a really special type of autoencoder called a variational autoencoder, or VAE. A normal autoencoder is basically just a good copy machine, but a VAE, it takes things a huge step further. It doesn't just learn to copy the data, it learns the underlying rules of the data, the recipe for how it's supposed to look. So it's not just copying, it's understanding.
And that is a total game changer for our missing data problem. Because a VAE understands the rules, it can make incredibly smart guesses about what a missing piece of the puzzle should look like. It can look at a patient's gene data and generate a totally realistic prediction for their missing protein data. That power is just unbelievably valuable.
And of course, the field hasn't stood still. Scientists have come up with even smarter ways to train these models. With adversarial training, you have two AIs compete. One acts like a forger, trying to make fake data, while the other acts like a detective, trying to spot the fakes. They force each other to get better. With contrastive learning, you basically teach the model to be a good matchmaker, to pull similar patients closer together on its map and push different ones further apart.
And the result of all this innovation? A complete explosion of new tools. Just since 2020, VAEs have become this dominant force in the field. We're talking well over 20 specialized models that have been built specifically to tackle the unique challenges of integrating all this biological data.
So where is all of this headed? Well, the next frontier is to look beyond just the ohms and start bringing in every single source of health information we can get our hands on.
This diagram really paints the picture of the future. We're moving towards what's called true multimodal integration. We're going to combine the molecular data with things like medical images, like MRIs or X-rays, and even the messy, unstructured notes from electronic health records. The goal is to build the most complete digital twin of a patient that we can possibly imagine. And the crazy part is, the technology to actually do this is already here. You've heard of foundation models. They're the AI architecture that powers tools like ChatGPT. Well, right now, those same kinds of models are being adapted for biology. Researchers are feeding them millions of biological samples to teach them the fundamental language of human health, so they can be fine-tuned to predict disease and revolutionize medicine. We are really just at the beginning of this journey. We are assembling the most detailed, high-resolution map of human health that has ever existed. From solving the multi-omics puzzle to building these incredible foundation models of biology, every single step is bringing us closer to that promise of truly personal, precision medicine. The only question left is, where will this map lead us next?